So in this part, I'm going to continue to take you through improvements that were made in microscopy technology to improve the resolution and the contrast of the images that you can obtain with these microscopes. An important person is George Airy, who was partially in Cambridge. Again, he was an astronomer, and he rigorously described the phenomenon of diffraction for the first time. So he showed that the far-field pattern of a plane wave illuminating a circular aperture of a size d results in a so-called Airy pattern in the far field that you can see here. So this is the far field, and you see the intensity distribution of the light if a plane wave comes onto the aperture. And so you get a pattern of circular rings with a bright central maximum, which is called the Airy disk. So this is the Airy disk here. And the first dark ring here that you see just on the outside of the Airy disk appears when there's destructive interference between the waves emanating from different parts of the aperture. So the Airy pattern, this pattern that you see here, this distinct pattern, is very important. In microscopy, this pattern is also called the point spread function, because it is exactly the image that you get when you image a point of light in the sample plane. So the image of a point in the sample plane in your microscope image will give you a pattern that's not a point, but it is an Airy pattern. And the position or the angle at which the first minimum appears, so this position here and this angle here, I'm sorry this wasn't very successful here. Let me just get rid of this this drawing. So the angle at which the first minimum appears, so this angle here, I don't know whether I can draw this easily here. This angle here is given by lambda over d, where lambda is the wavelength of light, and big D, capital D, is the diameter of the aperture. So the smaller the diameter of the opening here, the larger the angle will be, theta, at which the first minimum appears. So the aperture size and the size of this ring pattern are inversely proportional to one another. Think about this in terms of Huygens principle and try and see why that might be the case. To continue with the history, I mean, this man here, Ernst Abbe, he was a real microscopy hero, and he was also a hero in many other ways. He was the first chairman of the Carl Zeiss Foundation, and he lived in 1840 to 1905 in Jena in Germany. And he was the first one to really use rigorous mathematical principles to describe image formation in the microscope, and he built fantastic microscopes. I mean, to this day, Zeiss is one of the big manufacturers of light microscopy instruments. And his famous formula of image resolution that we will derive in this course is engraved on his tombstone. You see it here. And it says that the minimum distance d at which 2 points can be distinguished from one another. So if you have a light point here and another light point here in your sample, can you distinguish these 2 points, or will they fuse into each other because of diffraction? Because you get these Airy patterns, and the Airy patterns are broadened out, and they start overlapping, and the light leaks into one another. And he derived a formula that the minimum distance that you can resolve two object points is given by the wavelengths of light lambda divided by 2 times n sine alpha, where n is the refractive index, which you can take to be 1 for the moment, and sine alpha is the half angle of light entering the microscope objective. So the larger this angle alpha, this of the capture angle of the light is that goes into the objective, the better the resolution, because the smaller d becomes, and the smaller the feature sizes are that you can resolve. For those who have worked with microscopes, they will recognize that this denominator here, n sine alpha, is also called the numerical aperture of the objective lens. To move on, I would like to introduce this man here, Camillo Golgi, to you, because one issue is improving image resolution, so the smallest distance at which you can resolve two objects from one another and distinguish them, so that's Abbe. But the other is also to gain contrast, and contrast is a different thing. You may well have good resolution in the instrument, but you may still not see the features that you have because the image may be contaminated by out of focus light and other things, and that reduces contrast. Camillo Golgi was an Italian pathologist who worked on the central nervous system, and he was able for the first time to see individual neurons. We did not know before a little more than 100 years ago that the brain consisted of individual interconnected neurons, because, you know, if you take a bit of brain tissue, it's just this white mass. You can't distinguish individual neurons in this white mass if you image them. And Golgi was able to visualize neurons in a microscope because he stumbled upon this the so-called black reaction, which consists in fixing silver chromate particles to the neurolemma. So this is the membrane of the neuron by reacting silver nitrate with potassium dichromate. And then what happens is you get a precipitate inside the neurons, which is black, and it makes their morphology visible. So on some of the neurons, you can see them, they appear here in this black color. And the ingenuity of this black reaction for imaging brain sections is that it is very inefficient. So you may say, why is that an advantage in this context here? Well, the point is that in this black reaction, only a very, very small number of neurons are labeled with this precipitate. The vast majority, maybe 1 in a million neurons is labeled black, but the reaction doesn't affect the other, the rest of the neurons, the vast majority of the neurons. And so this is in fact why Golgi was able to see these individual neurons, and this is central to obtaining good contrast and resolution in microscopy to this day. The principle of optical super-resolution is based on the same principle. So please remember this concept. The idea here is to suppress the signal from most things in your sample in order to bring clarity to some individual things. So some of these neurons you see with exquisite detail, and the only reason why you see them with this exquisite detail is because you suppress the signal from all the other neurons in there. And this principle of suppressing the signal from almost everything except from a few things is what makes so-called super-resolution methods possible, for which people won a Nobel Prize a few years ago. So the father of neuroscience is this man here. He made these wonderful drawings of neurons. This is Santiago Ramón y Cajal. He obtained images like this from brain sections with Golgi's method, and these beautiful tree-like structures you see here are so-called cells of Purkinje. So we really understood how these neurons are connected by using this method in microscopes. And he established a so-called neuron doctrine, which is namely the idea that the central nervous system, the nervous system consists of discrete units which are interconnected. And funnily, I mean, he shared the Nobel Prize with Golgi in I think in 1906. Yes, in 1906. And in fact, they couldn't stand each other. These two scientists. They had very different personalities. And in fact, interestingly, although he came up with the black reaction, Golgi did not believe in the neuron doctrine, although, of course, Cajal was right. So we can see from Abbe's formula that the minimum resolution that you might get with an optical microscope may be of the order of a wavelength of light, lambda or so. We see this from diffraction. So we also saw this from the Huygens slit experiment. So you make the aperture smaller than the wavelength of light, and then you lose resolution. So the best we can hope for is to obtain an image with an optical microscope of the order of a wavelength of light, lambda. How do you go beyond that? You know, a protein is much smaller than that. A typical wavelength of light, 500 nanometers or half a micron in size. A human hair is 50 micron in size, in diameter. So you get some idea of scale. So how do you go below that? I mean, a protein, what's the size of a typical protein? A typical protein is of the order of 1 to 5 nanometers, perhaps. So how do you get anything that informs us on that scale? I mean, if you want to see how two proteins interact with one another, of course, a wavelength of light is too low in terms of resolution to be able to see that. We need to go below that. And so one way of obtaining information at a scale that's below the wavelength of light is through a technique called Förster resonance energy transfer. So Theodor Förster is this man here, shown on this photo. He came up with this idea of Förster resonance energy transfer, or FRET, which some of you may know about. And you know, I will go into this in detail, but essentially what you do with this method is you take two proteins which when they come together give you a signal, and only when they come together do you get that signal. And so obtaining the signal conveys information that is relevant to scales that's smaller than the wavelength of light, because you only get this when the two proteins bind to one another. And so this is one of the first methods that you can think of as beating the so-called diffraction limit to gain information below a wavelength of light. So it's an indirect method. It's not performing. It's not producing an image as such that is better resolved than a diffraction limited image, but it rather uses a spectroscopic reporter to give you a signal, and that signal informs you on something that is smaller than the wavelength of light. We will go into this in a separate lecture. So gaining resolution, improving contrast by suppressing things, these are tricks that are important. So we hear a bit about contrast, we hear a bit about resolution. But one other thing that you require is, if you want good contrast, you need to be specific to look only at those things that you're interested in. And so for that, we have today the technology of fluorescence microscopy. And this man here, Gregorio Weber, is often called the godfather of modern fluorescence microscopy. So he was an Argentinian scientist who briefly was in Cambridge, but he was mostly, in fact, in Sheffield. And he was able to synthesize a large number of dyes, and those dyes he could make so that they would stain specific parts of the cell with high specificity and with high selectivity. So, for example, he synthesized dyes like dansyl chloride, which bind to primary amino acid groups, and this can be used to label proteins in the cell. He was the inventor of what is today called immunohistochemistry. So what this means is that you label individual proteins in the cell using specific antibodies. So these antibodies will bind only to the class of proteins that you are interested in, and by putting fluorophores on these antibodies, you get signals only when these antibodies, only of the proteins that these antibodies bind to. And so you get these beautiful, colorful images that really allow us to see things inside the cell with high detail. So here's a molecular structure of diacetyl chloride, which actually is blue, and this I'm sorry, I got this wrong. The blue dye that you see on this image here is a dye called Ernst, which is also something that he derived, and it intercalates into DNA inside the cell, so it will locate to the nucleus where there is a lot of DNA, and so you see the nucleus here appearing in blue. Okay, so we've heard in the last part about how to improve resolution and how to improve contrast by suppressing signal that you don't want. And one further method, a very important method to reduce contrast, to improve the contrast and reduce out of focus light is the confocal microscope. And you must have seen confocal microscopes in different contexts. They're everywhere in laboratories all across the world. The confocal microscope was invented by this man here, Marvin Minsky, who was the founder of the MIT Artificial Intelligence Laboratory. And you see here a drawing of the patent application that Minsky put in 1961 for the confocal microscope. But in fact, the first units that are useful or resemble the commercial units you can buy today were built here in Cambridge by two scientists, Brad Amos and John White, who were at the LMB here, the Laboratory of Molecular Biology here in Cambridge. And the idea is to reject the out-of-focus light by using a pinhole that admits the light from the focused plane in the sample, but rejects all the other light from out-of-focus plane. And that really increases contrast and allows you to image at depth. We will go into this at a later lecture. But the confocal microscope is a key instrument to improve contrast and reduce out-of-focus light. The next part of microscopy history I want to talk to you about is the invention of the so-called green fluorescent protein, and this led to a Nobel Prize by these three gentlemen here, Osamu Shimomura, Martin Chalfie, and Roger Chen, and they shared the Nobel Prize for this truly great invention for the discovery and development of the green fluorescent protein, which essentially is a protein that is made by a jellyfish that lives in the North Atlantic, called Equoria victoria. And it turns out that this protein looks like a Coke can here. Here's a cartoon rendering of it, but in the middle of it, in the inside of it, there's a chromophore that looks like this, and this chromophore can be excited with light, and it emits intense green light. And these scientists have made it possible through genetic engineering, through biotechnology, to label specific proteins inside the cell using these green fluorescent proteins, using genetic encoding. And we will talk about that also in a later lecture. And that made it possible now to see inside living cells. We go back and think about the immunohistochemistry that was invented by Gregorio Weber earlier on. That required killing of cells. You couldn't just paint them with dye and image them. You have to kill them for those immunohistochemistry techniques to work. But these techniques use genetic engineering to get the proteins that you are interested in labeled, and that means you can watch them in living cells. The cells go about their normal business. So here you see images obtained, microscope images of two cells. So here's a cell. That's one cell here. So here's one cell, and then there's another cell around here, and you can see how these proteins are communicating from this cell to the other. You can see how they come to the cell membrane here, and the protein we're looking at is the so-called connexin protein. And the thing that I want to bring home to you is that the green color here tells you the concentration of that protein and just that protein. The green fluorescent protein will label only the protein that you engineer to be labeled, and that gives you incredibly high selectivity, specificity, and great signal, and the cell doesn't seem to mind. It goes about its business as in a normal way. So we will talk about GFPs also, of course, in a later lecture. So the final big invention was by these three scientists, Erich Betzig, Stefan Hell, and William Murnau, who shared the Nobel Prize in 2014 for the development of optical super-resolution methods which break the Abbe diffraction limit. So here, these are not indirect methods like Förster to gain super-resolution. These are real imaging methods that will produce in images that are more highly resolved than what you would get with normal diffraction limited images. And that really opened the door for us to observe life today at the molecular level. So you can see results here from our own laboratories. These are protein fibrils that cause Alzheimer's disease. And you see in the past you could obtain images like you see here in this top right-hand corner of the image, and then the super resolution improved the resolution to about this level here. So you can see a much better, clearer picture of life. So an animation of this is shown here. So we're looking at pictures, fluorescence microscopy pictures of amyloid beta. Amyloid beta is a protein that forms fibrillar structures that are associated with Alzheimer's disease. And so this is a resolution. So you see here 500 nanometers, half a micrometer in size is the scale bar here. And this is the best you can do with a normal diffraction limited top of the range microscope. But using the techniques of Betzig, Murnau, and Hell, you turn images like that into images like this. And you can see the vastly improved, not only the resolution, but also the contrast of the image that you gain using super resolution methods, for which the Nobel Prize was only awarded about 6 years ago. So you see it's a fascinating field. You can see why I'm fascinated about it, because we can look now at life at the molecular level. And with that, I finish this part of the lecture, and we'll go into more details in the next lecture on building the framework for what we need to understand how microscope images are formed and how we obtain the best possible performance from instruments.